Dear students, today we are going to study about microbial growth. Microbial growth is the increase in the number of cells. As the microorganisms grow, they undergo continuous cell division and the number of cells will increase, and that is how the microbial population is maintained. So the microorganisms, when they grow, they synthesize macromolecules from monomers by polymerization reaction, and these macromolecules, they will get accumulated in the cytoplasm of the cell and they will be assembled into new structures like cell wall, cytoplasmic membranes, etc. And as these cellular structures are synthesized, the bacterial cells, they will divide to produce new cells, and the bacterial cells, they divide by binary fission. Here in this picture you can see the parent bacterial cell. It will first undergo DNA replication so that two DNA strands are formed, and meanwhile other cellular components are also synthesized and the cell will enlarge and elongate, and a septum is formed in between the two bacterial DNA strands, and finally the cell will get pinched off into two and two daughter cells are formed. And when a cell divides into two daughter cells, we say that a new generation is formed, and the time taken for the formation of a new generation is called generation time. And this varies for different species of bacterial cultures, and for E. coli, under the laboratory conditions, if we are providing optimum nutrient conditions, the generation time of E. coli is about 20 minutes. And if the pattern of microbial growth follows a pattern that is the number of bacterial cells are doubling at regular time intervals, then it is called exponential growth. So in the case of E. coli, at every 20 minutes, if the conditions are optimum, at every 20 minutes the number of bacterial cultures will double. And this pattern of growth where the number of bacterial cells double at regular time intervals is called the exponential growth. Now let us see how the measurement of microbial growth can be done. The microbial growth can be measured by taking microbial cell number or by measuring the microbial cell mass. Microbial cell number can be counted by total cell count or by taking viable cell count. Total cell count can be done by microscopic methods or by doing flow cytometry. Microscopic cell count can be done for dried cells as well as for bacterial cells in liquid samples. Viable cell count can be done by spread plate method and pour plate method. Then microbial cell mass can be measured by taking dry weight or by doing turbidimetric methods or by measuring metabolic activity. Now let us see the details. The measurement of microbial cell number can be done by taking total cell count or by taking viable cell count. In the case of total cell count, we are counting the total number of cells in the sample, irrespective of whether they are live or dead cells. Both live and dead cells are taken into consideration in the case of total cell count, and this can be done by microscopic observation or by doing flow cytometry. Microscopic cell count can be done for dried cells and for bacterial cells in liquid samples. Dried cells can be stained and they can be observed under the microscope, and the number of cells can be counted. Then in the case of liquid samples, we use a special counting chamber called Petroff-Hauser counting chamber. It is similar to the counting chamber that we use for the enumeration of WBCs. This chamber, it is having a shallow cavity where we can fill the bacterial sample, and this cavity, it is having grids, and a cover slip is placed on top of the of this chamber after adding the bacterial samples. So the bacterial samples suspension will get filled in this cavity by capillary action. Then it is observed under the microscope, and as you see here, the bacterial cells will appear in this grid, and the number of cells in each cell or in each square is counted. So here we count both live cells as well as dead cells. Then in the case of flow cytometry, this technique is adopted for, it is used for counting the number of bacterial cells in liquid samples. So the bacterial cells in liquid samples, they are allowed to flow across a beam of laser or a beam of light, and as the bacterial cells, they pass through this laser beam or the light beam, they will be scattered, or if the bacterial cells are stained, they will emit fluorescence. So the fluorescence that is emitted from the stained cells or the light that is scattered by the cells, they are detected by a photodiode or a photomultiplier tube, and these signals are then converted to electrical signals which are then monitored by which are then analyzed by a computer, and that is how the number of cells in the samples are counted. Here in this case we take into consideration both live and dead cells, that is total cell count. Then the measurement of microbial cell number by viable cell counting or plate count. Here in this case we take into consideration only the live cells, and it is assumed that the live cells will undergo cell division by binary fission, and all the daughter cells which are formed from a single parent cell will adhere together forming a colony, and we are counting the number of colonies, and it is assumed that each bacterial colony is formed from a from a single parent cell in the original sample, and the data is expressed as the number of colony forming units, and the viable cell count is done by spread plate method and pour plate method. And before doing spread plate method or pour plate method, we have to first dilute the we have to first do serial dilution of the sample, otherwise if we are using undiluted sample, as you see here, the bacterial growth will be overcrowded, so that it is impossible to count the individual bacterial colonies. So in order to facilitate the counting of bacterial colonies, we have to first dilute the sample serially. For the serial dilution, we are taking 1 ml of the sample from the stock sample. It is added to 9 ml broth, so here it is getting 10 times diluted, and 1 ml from this diluted sample is added to another 9 ml of the broth, so that now the dilution is 100 times, and again 1 ml is taken from this and added to another 9 ml, so that the dilution is now 1000 times, 10 raised to 3 times dilution. So likewise serial dilution is done until we get pure bacterial colonies which can be counted, and the number of it is assumed that each colony is formed from a single bacterial culture in the original samples, and the colony count is multiplied by the dilution factor. So this diluted samples are now used for inoculation in spread plate method and pour plate method. In spread plate method, a small amount of the bacterial sample is added on top of an agar medium in a Petri plate, and with the help of a sterile glass rod, this bacterial sample is spread on the surface of the agar medium. Then it is incubated, and the bacterial colonies will grow on the surface of the agar medium, and the number of bacterial colonies are now counted and multiplied by the dilution factor. That is spread plate method. And in pour plate method, the bacterial sample is added to, a small amount of the bacterial sample is added to molten agar medium. It is mixed with the molten agar medium, and this mixture is then poured onto a Petri plate, and it is now allowed to solidify, and it is then incubated. So the bacterial colonies grow both on the surface of the agar medium as well as within the agar medium. Here also the number of bacterial colonies are counted, and it is multiplied by the dilution factor. So in this case, in both the spread plate method and pour plate method, we assume that the number of bacterial colonies correspond to the number of pure bacterial cells in this original sample. So the data is expressed as colony forming units. Then the next one is the measurement of microbial cell mass. The microbial cell mass can be measured by dry weight method, then by measuring metabolic activity, or by doing turbidimetric methods. Dry weight method is most suitable for measuring the growth of fungi. The fungi, as they grow, they will produce mycelia, and this mycelia can be collected by centrifugation. They can be washed to remove the adhering substances, then they can be dried in an oven and weighed. And if the dry weight, after the drying, the sample is weighed, and if the dry weight is more, that indicates more growth of the fungi, and if the dry weight is less, that indicates less growth. And this method is not suitable for bacterial cultures, since we cannot obtain sufficient quantities for measuring the weight after drying the samples. So it is not suitable for bacterial cultures. Then metabolic activity is another method by which we can measure the microbial cell mass. It is an indirect method. As the microorganisms grow, they will do metabolism, and certain compounds will be produced as a result of this metabolism, and they will be accumulated in the cell, and they can be measured. And these substances include organic acids, then oxygen consumption can be measured, then total protein can be measured, then cell wall constituents can also be measured. For example, in the case of fungi and filamentous bacteria, actinomycetes, N-acetyl glucosamine is a constituent in the cell wall. When the actinomycete cells undergo cell division, more cells will be formed, and the cell wall constituents, the amount of cell wall constituents will also be more. So the increase in the amount of N-acetyl glucosamine indicates an increase in the growth of this actinomycetes. So the amount of these substances produced, that is proportional to the magnitude of the bacterial population. If the amount of substances that are accumulated is more, that indicates a more bacterial growth, and if the amount is less, the bacterial growth is less. Then the next is the turbidimetric method. In this method, we measure the optical density. The turbidimetric turbidity is measured with the help of a spectrophotometer. We place the bacterial sample in liquid suspension in front of a light source, and the light rays, they pass through this bacterial samples, and as the light rays pass, when they hit the bacterial cells, they will be scattered, and the unscattered light rays that are emerging from the suspension, they are monitored, they are detected by a detector, and they are then measured. And if the number of bacterial cells is more, then the unscattered light that is emerging will be less. So that is how we measure the turbidity of the sample. And the commonly used wavelength for bacterial turbidity measurement is 600 nanometer, and the unit of turbidity measurement is optical density at the wavelength specified. For example, if we are taking the optical density measurement at 600 nanometer, then the unit will be OD 600. So an increase in the absorbance at a specified wavelength will indicate an increase in bacterial growth. That means the emerging light will be less. That means that the other light rays are either scattered or they are absorbed by the bacterial cells. So if the bacterial cells, if the absorbance is more, or if the emerging light is less, then that indicates the a profuse growth of bacterial cells. And if the absorbance is less, or if the emerging light is more, then that indicates a less growth of bacterial cells.